let's look at some important characteristics of enzymes. Enzymes are important proteins which speed up a variety of chemical reactions such as photosynthesis, respiration, and protein synthesis. They are biological catalysts and they lower the activation energy of a reaction. Their presence does not affect the nature and properties of any end product, and they remain unchanged during a chemical reaction. They work in different ways, but they are highly specific for their substrate. Some enzymes break down and some enzymes build up molecules, while some change one molecule into another. Now, how do they work? Enzymes work in conjunction to a substrate. Enzyme and substrate molecules are always moving, and when they collide with each other, the substrate fixes into the active site. This is called the collision theory. Now, in this theory, the enzyme and substrate must collide with sufficient energy and in certain orientation for a reaction to occur. The active site of an enzyme must fit into the substrate just like the lock and key. Once they fix together, the reaction takes place. Now the product is produced and the enzyme after that releases itself. In this example, the enzyme is breaking down the substrate into two products. Likewise, we also have enzymes such as proteases that break down protein into amino acids, or we also have carbohydrates which break carbohydrate into glucose, or we also have lipases that break down fats into fatty acid and glycerol. Hydrogen peroxide is a reactive oxygen species which is produced as a byproduct by the cells. Now we have enzyme catalase. It catches the hydrogen peroxide and breaks down it into water and oxygen molecule. As I told you, enzymes can build up molecules. Following the same principle, now enzymes are also sensitive proteins. They work best under certain conditions. First, there need to be a certain concentration of substrate for a reaction to be catalyzed. If the concentration of the substrate is low, the rate of reaction is going to get slow. Similarly, if there is a lot of substrate present, the reaction becomes slow because there would be less chances of the enzyme to bind with the substrate. Enzymes also need an optimum pH and a temperature condition to work efficiently. To a certain point, an increase in temperature increases the rate of reaction as there is more heat energy, and more heat energy means more collision. However, after a certain point of temperature, the rate of reaction starts to decreasing. But as you all know, because the enzymes are also proteins and they are going to denature. We are going to discuss the effect of pH, temperature on the enzyme in a detailed lecture about denaturation of the enzymes. Now, pH at which the enzyme works best depends upon the enzyme where it is present. For example, if the enzyme is present within the stomach, it is going to work best at a lower temperature as compared to the enzyme which is present in the saliva or the blood. Similarly, there need to be enough amount of enzymes to be around to speed up a chemical reaction. So, the important characteristics of the enzymes, let me just summarize them. These are that the enzyme are going to fit into their substrate exactly like a key in a lock. And after the reaction, the enzyme is not going to, there's not going to be any change in the enzyme, it's going to stay the same. Enzymes are sensitive to pH and temperature. For a reaction to occur, a sufficient amount of enzyme and substrate is also required. The enzyme control the rate of many biological reactions, but we also use them in our daily life. For example, the protease enzyme and the lipases enzyme, we use them in the laundry detergent, and these remove the protein. Enzymes within the laundry detergent remove the protein and fat-based stains on our clothes. Similarly, we also have enzymes which are used in the fruit industry, which break down the fruit cells and release the juice. Now let's look in detail about enzyme denaturation. Now we know that there are certain conditions under which the enzymes work best. In other words, it is the point at which the rate of reaction is highest. Among these points, the most well known are temperature and pH. If these conditions change from the optimal level for that enzyme, the enzyme can become denatured and consequently the rate of the reaction will decrease. Different enzymes have different optimal conditions. One result of enzyme denaturation can be disturbance in any balance system. For example, the homeostasis of different reactions in our body. For example, the activity of protease. If is not optimal, and it is changed in some way, it can affect protein digestion in our body. If we look in more detail, the denaturation of an enzyme or any other proteins means that the shape of the enzyme has changed in some way. Most of the time, the change is permanent. The activity of an enzyme depends on its shape, particularly on the shape of its active site, which binds to the substrate or a group of substrate. This change of structure means that the active site is not shaped for optimal binding rate of reaction. So, for example, if the temperature of the environment is too high for the activity of a specific enzyme, it may become denatured. Typically, the enzymes that are present in warm-blooded animals tend to have an optimal temperature of 37 degrees, which is also our body temperature. So, when the temperature rises above 37 degrees, the rate of reaction starts to decrease rapidly because the active site has changed its shape. Whereas in the enzymes which are present in the bacteria that live in thermal vents have much higher optimal temperature. Now let's see and understand the chemistry behind this change. The individual amino acids that make up the enzymes vibrate at a higher frequency at a higher temperature, which lead to the breaking of the hydrogen bond between the amino acids and different parts of the protein chain, leading to a change in its shape. At even higher temperature, the bonds between the amino acids, the peptide bonds, also break down, which leads to protein fragmentation or formation of protein fragments. Another important variable is pH. A pH which is higher or much lower than the optimal pH of an enzyme can change the shape of an enzyme and also its active site. Different enzymes have different optimal pH. As I told you earlier, that the optimal pH of the enzyme depends upon where that enzyme actually works. I also gave you the example of pepsin, which is an enzyme which is present within the stomach and it is involved in digestion of the protein. It has an optimal pH of 2. Now, how does the pH changes or affects the active site of an enzyme? If the pH is low, there would be a greater concentration of hydrogen ions in the environment than the normal ones. These greater number of hydrogen ions are going to interact with the amino acids, changing the shape of active site, and thus the activity of enzyme is going to reduce. So we can say that enzymes have optimal conditions in which they can work. Different enzymes have different preferred conditions of temperature as well as pH, under which they are able to perform their activity or catalyze the rate of reaction in a good manner.